So I'm going to start here. So it's my pleasure to welcome Dr. Ziegler. She's currently a senior investigator in the Office of the Director of the Epidemiology and Biostatistics Program of the National Cancer Institute. I've known her a long time. We've been colleagues and we've been working on multiple interesting projects. And so I'm very glad that she can come today and talk to you about some of the research she's doing on circulating vitamin D and the risk of colorectal and breast cancer in this very, very large consortium. This has been a multi-year effort on her part, as well as a large number of investigators. Just a little bit about her background. She's clearly well established. She actually started her training. She completed a BA in chemistry and English from Swarthmore College, and then she did a PhD in biochemistry from the University of California at Berkeley, and an MPH in epidemiology and public policy from the Harvard School of Public Health. She's taught numerous courses. She's published extensively. She's collaborated worldwide. And her major areas of research, it's hard to put it all together, but has been basically in nutrition. She's done a lot of work in vegetables, fruits, and carotenoids. She's done work in one-carbon metabolism, and clearly also in vitamin D. And then she's extensively mentored both, I think, undergraduates, postgraduates, as well as doctoral students and junior faculty. So it's my pleasure to welcome Regina Ziegler today. Thank you.
Thank you. I'm very pleased to be here, particularly because my daughter in the audience is about ready to graduate from Hopkins with her MD, PhD. I'm not used to using a stationary mic, so if I move away from it and you can't hear me, wave your hand and I'll try to come back to where the mic is. The sources of vitamin D vary around the world. In the US, the sources are diet, particularly fortified milk and fatty fish, also multivitamins and vitamin D supplements, and sunlight. Now, sunlight basically converts 7-dehydrocholesterol in the skin to cholecalciferol, using UV radiation. The cholecalciferol is hydroxylated in the liver and in other adipose tissue, forming 25-hydroxyvitamin D, which is the accepted measure of vitamin D status. Then there's another hydroxylation in the kidney and in other tissues to form the active form of vitamin D, which is 125-dihydroxyvitamin D. We don't actually measure 125-dihydroxyvitamin D because it's so tightly regulated by parathyroid hormone, calcium, and phosphate. Instead, we use the 25-hydroxyvitamin D as a measure of vitamin D status. The accepted function of vitamin D is bone health. It maintains circulating calcium and phosphate for bone growth and remodeling. It prevents rickets in children, and it reduces risk of osteoporosis in adults. But today we're going to talk about vitamin D and cancer, particularly if I go in the right direction. Okay, so vitamin D and cancer. Vitamin D is a fat-soluble vitamin, but it's also a steroid hormone. It binds to its nuclear receptor, VDR. It modulates cell proliferation, differentiation, apoptosis, and in fact, the VDR complex may influence the expression of 3 to 5% of the human genome. There are also effects of vitamin D that may be independent of the receptor, possibly related to immune function and inflammation. There is both experimental and epidemiologic evidence that vitamin D is involved in carcinogenesis. The evidence is most suggestive for colorectal cancer, and breast cancer, though none of the randomized trials of vitamin D supplementation have been conclusive. However, randomized trials do have some issues. They can not be using the right dosage. They can have too short a duration. The vitamin D supplements could have been administered for too short a time, or at the wrong period of life. And you also have a compliance issue. So a positive result from a clinical trial is impressive, but no results you have to be a little careful about. When we talk about vitamin D and cancer, we have to keep in mind that there are some concerns that there may be increased cancer risk for some cancers at high, particularly high 25-hydroxy vitamin D, specifically pancreatic cancer. In 2010, the Institute of Medicine made a report on the dietary reference intakes, the DRIs, for both calcium and vitamin D. And on the basis of that, they came up with what they thought were the appropriate dosages of circulating vitamin D. The IOM guidance was based solely on bone health. And that was because the experts concluded that the existing data on cancer risk in vitamin D, although provocative, was insufficient as a basis for the DRI. More research was needed. There was not a clear dose-response relationship. So what IOM said on the basis of bone health is that deficiency was less than 30 nanomoles per liter, and I'm also giving the units in nanograms per mil. Then we have the range for inadequacy, 30 to 50. 50 to 70 was considered adequate, again, for bone health. Greater than 70 was beyond adequacy. And there were concerns about adverse effects greater than 125. And there was concern about calcification in the tissues and in the blood vessels, and the implications of that for heart health, and kidney health. IOM did not establish an optimal 25-hydroxy range for all health. And as recently as 2016, the World Cancer Research Fund and the American Institute for Cancer Research said there's really only limited evidence for the role of vitamin D in colorectal carcinogenesis. Nope. Okay. I'll be talking about the first pooled analysis of vitamin D status and risk of breast and colorectal cancer. A pooling study actually works with data from individual study participants. It's more informative than a meta-analysis of summary results from each study because it's actually working with data from each individual in each participating cohort. The strengths of a pooled analysis include improved power, many more subjects, leading to more precise and robust findings. You can look at what's going on in population subgroups. You can look at what's going on for tumor subtypes. You can access a wide range of vitamin D exposure because you're bringing together so many different cohorts. And among these cohorts, there are diverse sources of vitamin D: sunlight, diet, fortification, supplementation, all of which affect the circulating 25-hydroxy vitamin D levels. However, there are some challenges. One, you have to harmonize the exposures, the covariates, the outcomes, and you need to use comparable statistical approaches across the study designs. But by doing this, you can eliminate the heterogeneity across studies that's present in many meta-analyses that simply rely on published results. Dr. Stephanie Smith-Warner from Harvard School of Public Health and I co-lead the vitamin D pooling project of breast and colorectal cancer. It was one of the earliest pooled analyses of circulating biomarkers and cancers, and we invited cohorts to participate through the Diet and Cancer Pooling Project and through the Cohort Consortium. Pooling the circulating 25OD data was particularly challenging. We wanted to use absolute concentrations. We wanted to get away from the traditional approach in which you look at high versus low, because high vitamin D in Norway could be very different from high vitamin D in Hawaii. And we wanted to use absolute concentrations and take advantage of the wide variation in circulating vitamin D around the world. So we wanted to leverage all the influence of vitamin D of geography, diet, fortification, supplementation, and lifestyle. Also, we knew we had to correct differences in 25-OHD measurement by assay, lab, and year. The assays for vitamin D, for 25-hydroxyvitamin D, are not great. There's a 20 to 50% variability across labs. And we want to use absolute values to facilitate translating our results into clinical guidance and to public health guidelines. As I mentioned, there's a wide range in the accuracy and precision for 25-hydroxyvitamin D assays for many reasons. But what we realized we needed to do for this study was to calibrate all of our vitamin D data, both the already collected data and the new data that we would collect, to a single respected assay. These are our participating cohorts. We assembled practically all of the circulating vitamin D data available worldwide for breast cancer and colorectal cancer. You can see that we have cohorts from North America, from Europe, and one from Asia. There are actually 21 cohorts, 3 continents, and 13 countries involved. You can see that for breast cancer, we have over 10,000 cases, over 13,000 controls. For colorectal cancer, we have over 3,000 female cases, almost 2,800 male cases, over 7,000 controls. The numbers in lighter blue are the, I want to get this straight. The blue indicates the studies for which we measured circulating vitamin D for our project. It had not already been measured, but we got funding to do additional measurements of vitamin D to increase the size of our study. The new assays that we brought in increased the breast vitamin D data by 16%. We added 5 new studies. But for colorectal cancer, the new assays that we conducted increased the available data by 43%. 8 of the 17 studies were new cohorts. This shows you the type of distribution around the world that we wanted to work with. This is data actually for the controls for our breast cancer studies, obviously all women. This is calibrated data, it's season standardized, and it's showing you the type of variation you can get around the world. The red lines represent the 10th percentile, the median, and then the 90th percentile. The green horizontal lines represent the IOM guidance. What you can see is that the median 25-hydroxyvitamin D is lowest in France and Italy. It's about 30, and it goes up to about 70 in the Harvard nurses cohorts, probably because of the vitamin supplements, and in Norway, probably because of fatty fish consumption and some fortification. And just looking at the range, you can see how it spans the IOM guidance. The median value for all 17 cohorts is about 50, which is the lower limit for vitamin D adequacy set by the IOM for bone health. All of this data is based on a common assay and season standardized. So it doesn't reflect differences in assays or lab or dates. It simply reflects differences in vitamin D exposure around the world. So this was our approach to calibration. For each individual study, and men and women were treated as individual studies for colorectal cancer, we reassayed replicate samples for 29 controls. It's a somewhat arbitrary number. About 3 were selected per decile. We wanted these controls that we reassayed to be representative of the 25-hydroxy vitamin D distribution in the controls for each individual study. We measured 25-hydroxy vitamin D by direct competitive chemiluminescence immunoassay at a widely respected lab, Heartland Assays. We used a single assay kit, a single manufacturing lot, similar laboratory methodology throughout the pooling project. For those of you who are familiar with vitamin D, the assay measures both the D3 form and the D2 form with equal responsiveness. Nowadays, we might have gone for LC tandem mass spec, but at that time it was too complicated, expensive, and untested. The assay performance was monitored with blinded QC. One issue the statisticians had as we started out was whether the imprecision due to calibration would limit our ability to see effects. And there was Mitch Gale did modeling, and the basic answer is that even though there was instability due to calibration, imprecision, it was less than the improved validity of calibrating all the data to a single assay. After calibration, 22% of the participants remained in the same consortium-wide decile, but 78% changed. This approach has now been adopted by several other pooling projects of circulating biomarkers. This is an example of one of our calibration curves. It's actually an impressive one because you can see that on the x-axis, the vitamin D for the original assay correlates really quite well for the vitamin D measure at the pooling project lab, Heartland's assay. It's a good correlation, and we used robust regression to calibrate all of the individual data, in this case in CPS2, based on this regression for 29 representative controls. This is another calibration curve. This is the opposite end of the spectra. The correlation is not as good, but again, we came up with the best line, robust regression, and calibrated the data. And the reason the correlation was not as good is that this study had originally used a different type of assay in a different lab. And so that's why it didn't correlate perfectly with the chemiluminescence assay at Heartland's. This was actually used the RIA in the I think in South Carolina lab. Actually in Bruce Hollis's lab. This shows you what happened after we calibrated the data. This is actually data for breast cancer controls plotted by cohort. If you see just a blue dot, this is a study where we didn't have to calibrate. It was new data in our lab. But where you see a orange dot going to a blue dot, that was the effect of calibration. The orange is the earlier data. The blue data is what happened after it was calibrated. Some medians went up, some medians went down, some changed a lot, some changed a little. So this is the data by cohort for the breast cancer controls. This is actually for the male colorectal cancer controls. The plot follows a similar logic, and again, you can see some values go up, some values go down. So it meant that the calibration was having an effect. I mentioned that we followed the laboratory performance with coefficients by a number of methods, but we generated coefficients of variation for NIST standards and for study-specific standards through all over 110 batches that were done. And in general, the QC, including both within batch and across batch variation, based on blinded QC material, was under 10%. Vitamin D presents a particular problem for biomarker studies in that there's seasonal variation in vitamin D. This is the nature of the seasonal variation. Again, we're looking at the male colorectal cancer controls. We are combining the data across all of the controls. It's calibrated data, and you can see how it varies by month of the year. It's the same type of plot we had before, where the red lines indicate the 10th percentile, the median, and the 90th percentile. The green horizontal lines reflect IOM guidance. And what you can see here is that the concentrations of vitamin D vary substantially due to sun exposure and intensity. In January, your values are running about 43. By the time you get up to August and September, the values are over 70. So you really have a huge impact of the season of blood collection on the circulating vitamin D levels. So this is how we adjusted for that. We ultimately standardized all of our vitamin D readings so that for each individual, they reflected that individual's average value over an entire year based on the seasonal variation in the community in which that individual lived. The problem is that in some studies, cases had bloods collected at one time of the year and controls at another time of the year. And while many studies match controls to cases on the general season of blood draw, they didn't match very tightly. And in addition, there was variation among all the studies, with some studies collecting blood more in the summer, and as in Norway, and others more in the winter. So we created a season standardized 25-hydroxy vitamin D value for each participant. What we did is for each individual study, we regressed 25-hydroxy vitamin D on the week of the blood draw using sine, cosine functions, and then each participant's residual difference from the predicted mean for the entire study was added back to get that participant's value. We also actually generated study-specific standardized vitamin D focused on week 9, which was a winter value, and week 34, which was a summer value. But today I'm just going to show you results for the all year round standardized vitamin D. This is an example of how we modeled 25-hydroxy vitamin D by week of blood draw. Each dot is an individual circulating vitamin D, and we fitted it by a 3-knot spline curve. On top, you have the Nurses' Health Study, which is centered in the US, and you can see there's not a lot of variation between winter and summer because this study includes people from all over the US. But values are higher in the summer than in the winter. Breakthrough Generations is based in the UK, the United Kingdom. There you start off with much lower values, and there's a much more obvious difference between winter values and summer values, because again, this is this is Great Britain. And what's interesting is you can see not only the higher values in summer, but there seems to be a tendency to go into the warmer climes somewhere around the middle of February, because you can see an increase in circulating vitamin D right at the end of February and at the beginning of March. Okay, now I'm going to show you our results for colorectal cancer, then I'll move to our results for breast cancer. The colorectal cancer paper has just been accepted by the Journal of the National Cancer Institute. So, the controls were selected by incidence density sampling. They were matched by study-specific factors to the cases. As I mentioned before, for colorectal, we measured pre-diagnostic circulating vitamin D for 8 cohorts as part of this pooling project, and then we calibrated previously collected data from 9 cohorts. And each participant's vitamin D was averaged over the entire year. That's what we mean by season standardized, according to the variation in that individual's community. The participant level data was harmonized centrally to create uniform definitions, because we're bringing in data from a lot of cohorts. All of the exposures were assessed prior to diagnosis of colorectal cancer. We worked with absolute 25 hydroxyvitamin D concentrations. We either categorized them by consortium-wide quintiles or by Institute of Medicine cut points. And we also modeled them continuously as 25 nanomoles per liter. We took a 2-stage approach to calculate pooled estimates. We calculated study-specific relative risk by conditional logistic regression, and then pooled the data using random effects models. We checked and found out that if we had used a two-stage approach with fixed effects models or an aggregated analysis where all the data is lumped together and it's a single one-stage approach, our results were very similar. So the analytic method didn't matter. Okay, we have some results now. This is the pooled relative risks of colorectal cancer by consortium-wide quintiles and continuous vitamin D. You can see the 5 quintiles arrayed across the top, and the first set of relative risks is just adjusted for the study matching factors. The second includes adjustment for BMI and physical activity, which are 2 confounders we worry a lot about. The next line is adjusted additionally for colorectal cancer risk factors. The last line is adjusted for dietary factors. Doesn't matter what model you use. The results are pretty much the same, which was encouraging because when you don't see a lot of confounding, it increases your confidence that you're looking at a causal relationship. We didn't have values bouncing all around. What you can see is that, now everything is relative to the middle quintile. Risk is significantly increased. It's increased by about 20% in the lowest quintile, and it's decreased by about 20% in the top quintile, again, all relative to the middle quintile. All of the trends are highly statistically significant. The relationship in women is actually stronger than that in men. And I'm showing you the model containing the colorectal cancer risk factors. The P4 trend in women is statistically significant. And this is a P4 trend based on the medians in each category. The P4 trend for men is not significant, but when we move to continuous variables, the relative risk per 25 nanomoles per liter, we see an inverse association of 0.81 in women which is significant, and 0.93 in men, which is at the edge of statistical significance. The upper confidence limit is 1.0, so we're almost there. It's not as if you're not seeing anything. There was definitely heterogeneity, and if you look carefully, but by sex, and if you look carefully at the data, you can see that the difference between the men and the women is at the very high vitamin D concentrations, not at the low vitamin D concentrations. Here we're looking at the pooled relative risks of colorectal cancer by the IOM-based categories. So we're talking about deficiency, insufficiency, sufficiency in terms of bone health, and we're seeing how the colorectal cancer risks compare. Try to look at the dark blue results, which are the overall results, and you can see that there is a 31% significantly higher risk among the people who have deficient vitamin D. And then the referent population is 50 to 62.5, which is the lower range of sufficiency for bone health. But for colorectal cancer, the risk continues to go down, such that at levels beyond sufficiency, 75 to 87.5 and 87.5 to 100, you have significant reductions in risk, 19% lower and 27% lower. We were particularly interested what would happen at very high levels, greater than 100 nanomoles per liter, and there was no evidence of ever increasing protection. The risk actually rises at the very highest level and becomes not significantly different from what it is in the referent population. Here we have a forest plot of the study-specific and pooled relative risk of colorectal cancer per 25 nanomoles of circulating vitamin D. And you can see that most of the results are inverse, whether you look at the men or the women. The effect in men, the pooled estimate in men of 0.93 is not as strong as the pooled estimate in women. But what I'd like to emphasize is as you look at all of these studies, only 5 of the studies, and they're all in women, were statistically significant. However, when we pooled the data, the results for the whole pooled study became highly significant, such that it was a p-value of about 10 to the minus 7, which is the advantage of pooling data. Now we're looking at the effects of colorectal cancer per 25 nanomoles of 25-hydroxyvitamin D in subpopulations. And all of the point estimates represented by the diamonds are less than 1. You have an inverse relationship in every single subgroup you look at, whether you stratify by age at blood draw, age at diagnosis, race, body mass index, physical activity, menopausal status, use of menopausal hormone therapy, or dietary calcium, and some other things that we'll get to on the other slide. This type of, and you can see that none of the P's for interaction were significant. So we're seeing a similar effect across all the subgroups. This is also, the consistency of this relationship is also evidence for causality. This is the same type of plot, and we're now looking at different subgroups. Again, note that all of the associations are inverse. None of the interactions are statistically significant. And here we see that whether the blood was collected in the summer or in the winter, we still had inverse relationships. We had it at all 3 latitudes that we looked at. We had it for the US and outside of the US. Whether we focused on calibrated data or newly measured data, still an inverse relationship. We saw similar, comparable, similarly strong inverse relationships for colon cancer and rectal cancer. And while it seemed as if the relationship for proximal colon cancer might be stronger than for distal colon cancer, the result was not statistically significant. So to summarize up our results for colorectal cancer, the colorectal cancer risks decreased steadily and significantly. Here's the p-value for the pooled continuous vitamin D exposure, 10 to the minus 7, with increasing pre-diagnostic vitamin D. All of the vitamin D was measured prior to disease. By demonstrating a strong, robust association, we strengthened the evidence which had been previously considered inconclusive for causality. And as I mentioned before, seeing little confounding, minimal confounding, and seeing effects across all subgroups was evidence that the relationship might well be causal. Deficient vitamin D, which we saw in 9% of the controls, was associated with 31% higher risk than the lower range of bone health sufficiency, which was in 23% of the controls. Risk continued to decrease at 25 OHD above sufficiency for bone health, was 20% lower for 75 to 100 nanomoles per liter, which was actually the optimal range in our study for vitamin D protection vis-a-vis colorectal cancer. Risk did not continue to decrease at concentrations above 100 nanomoles per liter. And it's important to emphasize this because some drug manufacturers and other groups are pushing for ever-increasing vitamin D intake. But we didn't have a lot of people with high levels. Only 4% of the controls had vitamin D at that level. So our optimal circulating vitamin D concentrations for colorectal cancer are 75 to 100, which are higher than the current IOM recommendations for bone health. Inverse associations were observed in all the subgroups, but risk reduction was greater in women than in men at high 25-hydroxy vitamin D. We don't know why. We know that you can see differences in the risk of colorectal cancer by BMI-related risk factors, by obesity-related risk factors. We know that in our study, we saw the same effects for premenopausal women and postmenopausal women, for postmenopausal women on exogenous hormones and those not on exogenous hormones. So the difference may not be simply estrogen, but clearly more experimental and epidemiologic research needs to be done on this sex difference. The other place where research needs to be done is on the relationship in minorities. We didn't have that many minorities in this particular study. And one needs to continue to look at the impact of very high values of circulating vitamin D, because while few of our people had very high levels greater than 100, these were values that were measured some time ago. And there's increasing use in some groups of supplemental vitamin D, and we need to know what happens with the increasing circulating vitamin D levels. On to breast cancer. Now, I should mention that Dr. Visvanathan is the lead on the breast cancer paper, and she's at Hopkins and in the audience. So the breast cancer methodology was pretty much the same as that used for colorectal cancer. We're looking at calibrated, season standardized data. We have more subjects, so we can look more closely at extreme vitamin D, either very low or very high vitamin D. Here we're just looking at the pooled relative risk of breast cancer by consortium-wide deciles. We don't have to use quintiles anymore. And as you can see, it is flat. There is no association. The P for trend is not statistically significant. And we're looking at levels, the lowest decile has concentrations less than 27 nanomoles per liter, and the highest decile has concentrations above 83 nanomoles per liter. So there's a pretty good range here, and we're not seeing anything. Now we're looking at breast cancer by the IOM-based categories of circulating vitamin D. And in this case, we actually went very high and tried to see what would happen at concentrations greater than 125 nanomoles per liter, which we had in 81 of our cases. Again, it's a huge study with over 10,000 cases. And the relationship is flat. The P for trend is not significant. There's no evidence for a relationship. So we tried one more time. We decided to see whether looking at values collected in the summer only, in other words, looking only at those participants whose blood was drawn in the summer, and dividing the summer vitamin D into deciles, looking at that data and looking at winter data. Again, only blood taken in the winter, and dividing winter values into deciles, we tried to see whether that would show anything, because people have speculated that what is important, or what may be important, is your vitamin D in the winter when it's very, very low. Or what may be important is your vitamin D in the summer when you're accumulating stores in adipose tissue to last you through the next season. Navy blue is winter, red is summer. Whichever you want to look at, we don't see anything. Both of the relationships are null. None of the P's for trend are statistically significant, and it's probably obvious, but the reason the blue values are to the left of the right values is just that circulating vitamin D is lower in the winter than it is in the summer. This is looking at a forest plot of the vitamin D data by the individual breast cancer cohorts. And we're looking at the relative risks by 25 nanomoles of circulating vitamin D. And the point estimate is 0.99, which is pretty close to 1.0. Only one of these studies, actually the Nurses' Health Study, which is the second from the bottom, was itself statistically significant. All the other breast cancer studies, some had positive associations, some had inverse associations. None of them reached statistical significance. CALA is in the midst of finalizing the vitamin D and breast cancer analysis. So far, there's been no evidence of confounding by BMI, by physical activity, by other breast cancer risk factors. We've talked about looking at alternative ways to explore season of blood collection. We have looked at timing from blood collection to diagnosis. We're looking at subpopulations. It's possible there may be something by tumor subtype. And this is very important because if you find an effect that isn't by chance alone in very aggressive cancer or very aggressive breast cancer or in triple-negative breast cancer, even though it's a small population in a very large study, you need to know whether that's real. And so that's what CALA is working on now. In preparation for this study, Mitch Gale at NCI and Molin Wong at Harvard did develop two statistical papers on approaches for pooling circulating biomarker data. And as I mentioned, this is one of the first studies to pool circulating biomarker data. Mitch's study has been published in Statistics in Medicine, if there are any statisticians in the audience, and Molin's paper has been submitted to statistics in medicine. When we initiated this study, the vitamin D standardization certification program, which is lovingly known as VDSCP, did not exist. So all of the vitamin D data that was used by IOM for calculating the appropriate ranges for bone health, and all of our studies just accepted vitamin D values as they came. There was no standardization to a common internationally accepted reference method. Since that time, CDC did develop a program that evaluates 25-hydroxy vitamin D assays, and many of them are bad. We used a very good one, but many of them are bad, that evaluates the assays relative to an internationally accepted reference method that's traceable to the National Institute of Standard and Technology and Ghent reference measurements. It's actually an LC tandem mass spec assay. At the present time, 8 national health surveys, including Haynes, is participating in this program. And in the last 2 to 3 years, 27 clinical and research laboratories in over 10 countries have decided it's really important to start participating. What we did so that we could participate is we sent 200 of our serum samples, which were representative of the 25OH distribution across all of our cohorts, to CDC. So they're standardizing them and will be able to adjust our actual values to the standardized values from CDC. We're going to do this by robust regression, and this will enable us to relate our findings to public health surveys and clinical results, because increasingly more labs and more national surveys are going to be using standardized vitamin D data. Just 2 more slides, because one question we've asked among ourselves is, it time for translation? We had set up this study using absolute concentration so that we could translate our results into public health guidance. And it's not, because the problem is that we're finding that higher values of 25-hydroxy vitamin D may be optimal for colorectal cancer risk reduction, but until one has a greater sense of how 25-hydroxy vitamin D affects a range of health outcomes in both a protective manner and an adverse manner, our guidance for colorectal cancer cannot be extended to every other cancer and all other diseases. The other thing that is ongoing is we very much wanted to coordinate with VITAL, which is the vitamin D and omega 3 trial. It's the largest randomized trial, placebo-controlled trial of vitamin D. 2,000 IUs, which is a lot of vitamin D, is being given daily to 20,000 men and women. The primary endpoints are cancer and cardiovascular disease. Vitamin D is measured at baseline, 1 year, and 5 years, and we have heard that results may come available this summer. We're not sure. What we worked out with VITAL was to take 150 of their serum samples that they were assaying by their method and to assay them by our laboratory method, so that our dose-response relationship could be related to their effect. So let's say at a certain concentration, they see a certain impact on colorectal cancer or on breast cancer, or no impact. We'll be able to take that concentration and see what it is in our study, because it's about time we started comparing pooling projects of prospective studies with the randomized clinical trials, rather than just saying, hey, they're different. The 150 replicate samples that we reassayed cover a wide range of dietary vitamin D and supplemental vitamin D, and they're going to facilitate comparing the 2 studies. And then finally, I would like to try to emphasize what the strengths are of doing pooling prospective studies of circulating biomarkers, because when one talks at a medical school, there's a tendency to believe that unless the result comes from a randomized clinical trial, it's meaningless. And as an epidemiologist, surprisingly, I don't see it quite that way. Among the advantages of our pooling project is that we included a broad range of exposure, much wider than you would get in a single randomized trial. We are looking at physiologically meaningful levels of exposure, what we really see in a wide range of populations. There's variation in the determinants of the exposure because the vitamin D is being affected by sunlight, diet, supplements, lifestyle. We have a huge number of cancer outcomes, many more cancer outcomes than one is likely to get from vital. And this gives us precise, robust estimates of risk. We can do subpopulation analyses. We can do tumor subtype analyses. And ultimately, because of the range of vitamin D, we can look at dose-response relationships. By harmonizing our participant-level data for exposures and outcomes and analyzing with a common statistical approach, we can remove the heterogeneity which is potentially present in meta-analyses, and by including the vast majority of the prospective data available worldwide, we can markedly reduce the potential for publication bias. Thank you. No, Gary? I mean, Gary's my cousin, so I have to ask him.
So, is there any thoughts about why there was a difference in the breast cancer and the colorectal cancer outcomes?
To be very honest, no. There are many, many thoughts. We've discussed it at the vitamin D workshops. We've talked with many of the people who are doing laboratory studies of vitamin D in vitro and in vivo. And people have speculated, but I don't think we really know. What we do know is that in the colorectal mucosa, you can pick up the VDR receptor, you can pick up the enzymes that synthesize, that metabolize vitamin D, that synthesize it. So you have all the things in colorectal mucosa that might make vitamin D important, but you have it in breast tissue also. It could be that what we're looking at is something related to immunity and inflammation rather than cell cycle regulation through the VDR receptor. But believe me, we've talked to, Dr. Flood, who I'm sure you don't know, but we've spoken to a number of people, and it's an important question because, to go back a step, if you look at all of the micronutrients that we thought at one time were related to cancer, very few have hung in there. Vitamin D is one that many people feel has hung in, but it's only for certain cancer sites. And why do we see the effect only for certain sites and not for other sites? I don't know.
Was there a difference whether they got the vitamin D from oral supplements or from sunlight, or?
We have not, I'm not sure we can totally look at that in our data. And just because of the quality of the data coming in, and so far we haven't seen anything.
Well, actually, I had a comment on this difference between the colorectal and breast. Did you look at the time between when the blood was drawn and the cancer incidence, and whether the farther out you got, you might have less of a correlation, whether that might influence the target site?
We did. I didn't go into that because this can be very complicated, but in both the colorectal analyses and breast analyses, we played two games. We excluded bloods that had been collected just prior to diagnosis because of the concern of preclinical disease, which, and both colorectal cancer and breast cancer are developing long before they become clinically apparent, didn't make a difference in the results. And we also stratified elapsed time to see whether that made a difference, like the people who were diagnosed 10 years after blood draw, 5 to 10 years, less than 5 years, didn't make a difference. But it's a good question.
Nice presentation. You had mentioned, though, an association between high vitamin D levels and pancreatic cancer.
Yeah.
So what's known about that?
One of my colleagues has found that to be true in what's called the ATBC cohort, which is a very unusual cohort of smokers living in Finland. To be very honest with you, I think other analyses that have been done more recently, not all of which have been published, are undermining the concern about pancreatic cancer. It was, again, the ATBC cohort is so unusual, and it did come up with this pancreatic cancer, increased risk of pancreatic cancer, but it's not holding up in other studies, which is why you always have to replicate results.
But I wonder if there's anything unique about the geography, where they're located. So if you wanted
okay.
to do, would you do another Scandinavian country or, I'm not
It
sure.
has been suggested that, vitamin D in Finland is quite low, which, could have an impact on various physiologic diseases or physiologic processes. It's actually been suggested that the pancreatic cancer people do a pooling project like we've done. Pancreatic cancer is hard because you just don't get, to get a result for pancreatic cancer, you almost have to bring in 3 or 4 or 5 cohorts. And if I remember correctly, last year, this was discussed as a important project to vote up or down this relationship and to try to pull data. I think the problem is some very large studies are not replicating the result. They haven't all come out. And so I think there's reluctance to start a big pooling project if what people know is in the pipeline may decrease the enthusiasm for this hypothesis.
Finally, just one other thing. What's the receptor density in the
I could
different
give
tissues?
you the paper. I could send you the paper if you want to.
What's the
original
receptor density in the different tissues, colon versus breast?
Wait, I'm sorry, I didn't.
Receptor density, vitamin D receptor density. In other words,
No, but it's a very good question.
yeah, does that have anything to do with tissue response?
I don't know.
Okay. Is this on? Okay. I have one more question, Gina. So we're getting different results for different cancers. Have you thought about how we are going to work out an optimal dose? So, for example, colorectal, we see a protective effect, breast we don't. Do we need to do more studies that incorporate multiple cancer endpoints to try to understand what would be the optimal dose? Or is it doing more single studies of different cancers? What do you think?
I think the first thing, and maybe I'm saying this because originally I got my PhD in biochemistry, the first thing we have to do is get on top of the vitamin D assays. The data in the literature is based on assays that vary, as I think I mentioned, 20 to 50%. In other words, you can take a sample and send it to various labs, and the results will vary by 20 to 50%. And with that type of imprecision and inaccuracy, we have to move beyond that. as important as the IOM guidelines were, the fact that they were based on a range of assays, some of which didn't even incorporate NIST standards, means we have to be careful. So I think it's important going forward to make sure that the labs we use for any of this epidemiologic stuff have been standardized, so we can start trusting the data and compare it. Then maybe the differences we see in dose-response relationships may not be as striking. That doesn't quite answer your question. I just think it's the next thing we need to do.
Great. Well, we're going to have to end there. So I want to thank Dr. Ziegler for presenting some great data. And thanks, everyone.